May of every year would present on what I was doing at Thermo Fisher at the time was targeted sequencing, which I'll go into in a little bit of detail. But I was telling Renuka that I really want to keep this simple and straightforward, and I don't want to go too deep, right? But I do want to give you and empower you with the technical tools that you may need to be, we'll just say inspired by the technology, but not overwhelmed. All right. So at the end of the presentation, if you want to ask me questions, I think that would be the best to kind of tailor this to the specific needs of each student. All right. So we decided to title it Recent Advances in Genome Sequencing, but I'm going to talk about a lot of different things that we do at Pacific Coast Genomics. And then Renuka asked me to share a little bit about my journey, which I will toward the end, and how I ended up kind of founding my own company and starting my own thing. In fact, was just talking to a colleague tonight that I worked with at Thermo Fisher, and he's in a similar position where he's looking at starting his own company too. So, life is a journey, and as you learn these technologies, especially as you're in college and you're studying, it's good to keep in mind, how are you going to apply this knowledge, and what are you wanting to do? What are your aspirations in your career, and what does that look like? So I think we'll make it multifaceted here. So I'll talk a little bit about my journey in molecular biology, and as Renuka said, Renuka and I actually met at Washington State University. I always found Renuka to be, she's a wonderful example and a great leader to follow. Okay, so I witnessed that firsthand working with Renuka, and we were in a graduate student group of about 39 students at the time. It fluctuated, but there was a lot of us together. And so these years while you're in college, they're great years to learn and study. We were all studying plant pathology together, but I was focused on molecular techniques in plant breeding and focused on wheat breeding at the time. And from there, I didn't put it in the slide, but I actually spent 2 years in Australia as kind of a postdoc. And I was working with the Grain Research and Development Corporation there, doing business development for a qPCR assay for soil-based pathogen testing. So we were testing pathogens for all the important crops that were grown in the grain belt, which would be anything from canola to wheat, and everything in between. So was kind of working with that test and trying to develop it commercially. And then in 2014, I came back to Washington and went to work for Syngenta, which was you may be familiar with them. They're a large multinational company, and I basically got back into wheat breeding, and at that point, I started to get into the business side of my career, and that was mending the molecular biology and the tools that I had learned and practiced in graduate school and starting to apply those to, developing products and releasing products into the market in terms of a private industry entity doing business, right? So you start to learn a bit of a different skill set in that, in interacting with customers and practicing science in that sort of a context. And then eventually I ended up relocating to California. And at that point, I wanted to take my career in a different direction. And Bayer, who was a competitor of Syngenta, approached me. And I spent 4 more years then working for Bayer within wheat breeding. But I always, at that point in 2017, when I relocated to California, I really had a desire to start my own company, but it wasn't time yet. And there were people that told me that, but I really had a desire to start my own company. And it was a good thing because I spent another 4 years practicing wheat breeding and doing more commercial type. I was a commercial manager, so I was doing a lot of the business side of interacting with customers and releasing varieties into the market. And then in January or December of 2021, I met a colleague who just called me tonight, and he worked for Thermo Fisher Scientific. And this is really where I wanted to take my career in that direction at that time. I was probably 45, 46, and I had been working in my career for 15 years, and I really wanted to go into the full-time molecular biology. I wanted to dive into sequencing, and I wanted to learn all the aspects of the depth of molecular biology. And Thermo Fisher, as you know, is a global company that is very influential in all of those technologies, right? So uh, during my tenure there, I did a lot of different things, but one of the things that I led was an effort to design pre-designed targeted sequencing products. And as I go into some of the sequencing technology, you'll pick up that some of it can be a little bit cumbersome. It can be, a little technical and difficult to develop and design. And so while I was at Thermo Fisher, I utilized the bioinformatics scientists and the people that I was working with in the AgriSeq team to make these pre-made products that people could basically just go out and test and run. And Renuka was asking me a little bit before the webinar on what is targeted sequencing. I'll get into it a little bit later, but it's basically taking targets throughout the genome, that is specific regions in the genome that we know carry important information. May be CRISPR type information, it may be just SNP based or variant type information, and we amplify those regions. We can go up to 5,000 markers or 5,000 targets that we amplify, and then we sequence them, right? And so that can be very powerful when you do that over thousands and thousands of samples in a breeding program. All right, so I spent about 3 years there, and then I decided to kind of go out on my own, and I started to I actually worked as a contractor for another company for about my first 6 months after leaving Thermo Fisher, but I started to do practice on my own. I started to do my own business, right? And I started to carve out what that looked like and what the demand looked like and what the revenue looked like and what the opportunities looked like, right? And that's when things started to kind of fall into place for me. And after doing that for about 6 months, I realized I was at a place where I could start my own company and kind of do my own thing. So in July of 2025, I formally started my own company, and that was doing targeted sequencing panel design based on the Thermo Fisher Scientific technology, and providing bioinformatics support for service providers who actually do these sequencing workflows. I also realized that one of the weaknesses in the market and one of the needs was the ability to extract DNA at a good quality level at a lower cost. So I started to develop some of my own chemistries. I understand Renuka mentioned that some of the people joining the call might have a biochemistry background, right? This is you get in the molarities of different sodium hydroxide and tris HCl and these things that go into nucleic acid extraction. You have to have a good handle on all that and developing your own chemistries. And so that's a lot of what I do at Pacific Coast Genomics. And then at the end of the day, our clients want to know whether markers amplify in their material, and they want to know specific targets of interest. And so I run a single marker qPCR based genotyping assay service. So it's kind of where I'm at now. That's my journey. That's kind of the track that I took, and my background. So in this webinar, I really want to, again, keep it simple, not get too detailed, but give you enough information, starting with what is DNA, right? I'm sure all of you know, 99% of you or 100% of you probably all know what DNA comes from, what is the basic structure of DNA. And then I'm going to talk a little bit about sequencing technology history, because as you go into learning about where sequencing is today, it's good to get an idea of where it came from and its historical context. And then I'm going to go into a little bit about what we do with DNA. All right. So what do we do at Pacific Coast Genomics with DNA? What do our customers want to do with DNA, and what can you do? So as you're studying and you're learning all these fundamental principles that may seem fundamental and simple, uh, later on in your life and in your career, you put those things to use, right? So you don't always think of that when you're spending time studying, but that's how it works. And then I'll go into a little bit about marker development and how sequencing works in marker development. And then how do we implement all of that into breeding programs? Because that's really what I do at Pacific Coast Genomics. We try to take highly technical information and sequencing type information and actually implement it into something that the customer can use. So you can't get very far in talking about the history of deoxyribonucleic acid, which is DNA, right? Without talking about the major award-winning uh, discovery of Watson and Crick in 1953. So in 1963, 10 years later, they were awarded the Nobel Peace Prize for discovering the double helix structure of DNA. Okay, so it's got a phosphate backbone. It's adenine and thymine in a double phosphate bond, and it's cytosine that always binds to guanine in a triple phosphate bond, right? So that discovery really laid the foundation for everything that we do now. The next point, which was also a Nobel Peace Prize award-winning discovery, was polymerase chain reaction, right? And I'm not telling you anything that you don't already know. These are the foundational principles of molecular biology, but I think it's really good to revisit all this. So Kerry Mullis from Cetus Corporation, he worked for a company in San Francisco, and he got this idea that he knew that when you heat DNA up to 94 degrees, it naturally unzips, right? And then they learned that as you cool it, somewhere between 50 and 65 degrees Celsius, these primers will anneal. And then as you heat the DNA back up to 72 degrees, you have extension by a molecule known as Taq polymerase. That was another major discovery. So this machine, all it does is heats DNA up and cools it to these temperatures. And as you add, you guys all know this, as you add the components of Taq polymerase, oligonucleotides, and the all of the chemistry that goes into the reaction, voila, you get amplified DNA after 35 cycles that you can actually visualize and see on a gel. Okay. So it's always important to go back to a gel. And honestly, I still use gels today in what I do. Even though we can do all this sequencing and we have all of this technology, I still have to go back and double check my DNA quality on a gel every now and make sure it's not smeared, which is a sign of DNA degradation, as you know, and make sure that certain things are present, right? So um, you guys all know this, but basically DNA is charged, and as you move it from negative to the positive charge with about 90 volts for a certain amount of time, you actually pull that amplicon through the gel, or it may be the genomic DNA. But smaller pieces move faster, further, and larger pieces don't move as far, right? So that's how we migrate things in a gel, and that's how we distinguish an amplicon size, which was a polymorphic tool. Now, at the time when Renuka and I were in graduate school, Renuka, that was already almost 20 years ago, right? Um, this was one of the main technologies that we used. We actually went another step to a CE, a capillary electrophoresis machine at that time, which will do Sanger sequencing, right? And I understand that you guys know a little bit about Sanger sequencing, but we used to use that instrument to look at fragment size. Okay, and that's how I did all of my molecular mapping and developed my chromosome maps for my PhD 20 years ago. All right. So it's always good to lay this foundation of history. Okay, so what's happening in let's just revisit all the fundamentals here. So what's happening in in DNA amplification? We've got a forward primer, right? You guys all know this, and a reverse primer, and we have our genomic DNA. And we have the primer that anneals to the complementary sequence, and we have a SNP, which is a target of interest. Okay, so Renuka was asking what we do with targeted sequencing. Well, this is exactly what we do. A lot of times we know with a reference genome, we know where this SNP is. Now, what is a SNP? A SNP is where you take, whether it's a human subject or a plant subject, you take two subjects and you align the sequences at that chromosome position and location, and you see everything else is conserved. So on the left of this sequence, you see in the individuals, GGA is the same as the reference sequence and GGG is the same, but you see this base pair shift between two individuals from a thymine to a cytosine. Okay, as you know, that's a SNP. So that's a difference. And without a difference in the DNA, we really can't do anything. For example, if we designed these primers and there was no difference between all the subjects that we test, we can't investigate a crime because there's no difference in the DNA. We can't really investigate whether two plant types are different because there's no difference in the DNA. But where we have the difference, that's what we want to focus on. We have to have that variant and we have to be able to characterize that variant. That's what we do with sequencing. Okay, so just taking this into practice, when we amplify with amplicon, let's just say amplicon sequencing, right? So we amplify this target with a PCR, we get a 200 base pair PCR product, and we have variety A of let's just say wheat, and we have variety B, and we have this SNP that we know is on chromosome 1B, okay? And we know it's at 170,000 base pairs. That's basically what we use, right? The position, the physical position on the chromosome. And we know that this SNP is there. That's everything that we do in like targeted sequencing or even whole genome sequencing that I get into. That's the basis for everything. We've got to know that SNP and we've got to know where it's at. And if we don't know what it is and where it's at, we can't do a whole lot. Now, we can do discovery, which I'll go into. I want to lay a little bit more of the foundation, though, of the historical context of sequencing, because a lot of people may not know that in the year 2000, when all we had available was a CE instrument and Sanger sequencing, it was very, very expensive to sequence the human genome. All right. So it was 100 million, right? And then by 2007, with the advances in sequencing chemistry and technology and instrumentation and advances in the technology, we got down to about 10 million. And then in 2008, we got down to about 1 million. And I'll tell you that Illumina was purchased by from another company in around 2006. So you see the advances in how quick Illumina made progress. And I do credit them with a lot of this progress. 2011, we get down to 10,000. And by 2022, we're at about $100 for a human sample. Now, with targeted sequencing, a human sample is still around $100 to $125. Let's just say for an oncology sample, if a medical doctor is wanting to test for the presence of cancer, that's going to be the pricing. But in agriculture and what we did within Thermo Fisher within the business, we had the pricing for agriculture and plant samples a lot lower than the human side of the business. So, we were down between $10 and $20 a sample. All right. So that's kind of the context and the difference in over time and how far the advancements of sequencing have come. So right there, I think I'll just stop, Renuka. And do we have any questions, or does anybody have any questions, comments?
Grant, I have to correct one thing. We did not graduate 20 years ago.
Uh,
It was 10 years, 14 years something.
Okay, 15. Yeah.
Just kidding, yeah. Anyway, students, you can ask questions. Otherwise, it looks, sounds we are too
Every
old,
year
right?
matters when you get to be our age, Renuka. But it was 20 years ago when we started.
Could be,
I
yeah.
started in 2006. So,
Yeah, yeah. Students, any
okay.
question?
Any questions, students? Okay. Well, I'll carry on here, and I'm hoping that this is beneficial and that you're getting something out of this. So as we start to look at the sequencing market size and all of the elements that are in that, right now we're sitting at about 17 billion for the NGS market size, um, in general, right? So about, and this is actually 2025 statistics, but about 30% of that is oncology, cancer research. 25% is clinical investigation. 20% is reproductive health. So reproductive health, one of the things that's in with it's a big component of reproductive health is if you've ever had IVF or had friends that had IVF treatment and you're testing the embryos for, different traits of interest, that would fall into that category. Immune system monitoring, metagenomics, epidemiology, and drug development, which is kind of the pharmaco industry. And then agrigenomics actually only makes up about 4%. And that's where I tend to function in my company. I do other things as well. I have looked into going into the reproductive health industry with targeted sequencing. I honestly, I just don't have time right now because everything that I do is focused on agrigenomic customers. And then just general consumer genomics is around 3%. This would be like your 23andMe and other things that a lot of animal testing is in this category where you want to test your dog for pedigree and that kind of thing. So if we look at the sequencing companies and the major players that are in this space, there's probably what I call a big five. There's Illumina with their next gen sequencing platform. Thermo Fisher has the Ion Torrent. Oxford Nanopore has their technology. The Element has the Aviti and the Vitari, and the Singular Genomics has their instrument. I believe they're one of their main selling instruments is called a G99. I have a client that is starting to experiment with one of those, and we're looking at developing targeted sequencing on multiple sequencing platforms. Roche and Bio-Rad are involved, maybe in selling everything from reagents to support kits and that kind of thing as well, as Qiagen and some of the other companies that are out there. But those companies that I put their they're basically their logo on are the main companies. BGI is another main company as well that's growing. Okay, so when we talk about what is sequencing, it kind of falls into different categories. So whole genome sequencing has been done in practice all the way back to 2006. And before that, I should have put Sanger sequencing on here. I'm sorry, I was focused on NGS, which is next gen sequencing, and I assumed that you knew a lot about Sanger already. But Sanger would be included in this as well. Sanger is not necessarily high throughput, but Sanger is very effective and it's very accurate at identifying fragments and sequences, and as you know. So whole genome sequencing is basically done usually with an enzyme-based genome fragmentation. So you basically digest the genome with enzymes because most of the sequencing technology that's out there and I should have put PacBio in their logo out on that uh, the last slide, because PacBio is responsible for large fragment genome sequencing. So that's really what they specialize in, are 1 KB and above fragments. But most sequencing platforms only sequence 150, a 200 base pair, or a 300 base pair fragment. We did have chemistry with the ion torrent where we could go up to about 400 base pairs. But if you want base pairs beyond that, you kind of have to. Illumina does have larger base pairs, but it's not really higher than 300 or 400. Uh, PacBio is really the go-to for larger fragments if you want to sequence larger fragments. And so you've got to digest the genome, or you've got to split it up into smaller fragments, whether that's with amplicons or with enzymes, in order to sequence those smaller fragments. Okay. And so with whole genome sequencing, the enzyme-based fragmentation is not necessarily targeted like it is with targeted sequencing, but it's comprehensive, right? So you're able to sequence all these fragments, and you can get up to 1 million variants or more. Okay, so whole genome sequencing is typically used for discovery. It's for discovery of new variants. That's how it was used years ago, and 20 years ago. I can say that 20 years ago. So, and it's used that way today as well. And it dovetails a little bit with the microarrays, where customers will move from sequencing, whole genome sequencing discovery, to a microarray type platform where they can put all of those SNPs and targets onto one microarray. There are limitations with that as well, but obviously a microarray is not sequencing, but the two, I include that on the slide because the two work hand in hand. Low pass sequencing is another form of, or application of sequencing, right? So basically with low pass sequencing, what we're doing is reducing the coverage with the reagents that we put into the sequencer. So we're still using an enzyme-based genome fragmentation, which is cheaper. It's a lot cheaper than amplicon sequencing, and it's effective, but it really relies on bioinformatic construction and alignment of those small fragments that are sequenced back to the reference genome. So you need to work with somebody like GenCove, or you need to work with another company that is able to do that alignment. And if any of you are heavy into bioinformatics sciences, this is a real opportunity. There's a lot of opportunity out there. And I've got another slide that summarizes this for what we call dry lab work, for bioinformatic work and alignment to reference genome. So if you can learn how to do this, I can tell you right now, these are some very high paying jobs in the industry. Okay. Low pass sequencing is a little higher sample throughput than whole genome sequencing, because basically you're using less reagents, you're sequencing less of a coverage. All right. Then we move to targeted sequencing, which is what we did with AgriSeq at Thermo Fisher, and what a lot of my customers still do, and I support them doing that now. Amplicon sequencing relies on a lot of bioinformatics up front. Okay, so basically what we're doing with amplicon sequencing or targeted sequencing is, I ask the customer, give me a reference genome, and give me and from that reference genome, I can get what chromosomes are which. Usually it's published in the reference genome. A lot of them are annotated that way. And then I can get the chromosome position of that particular reference genome, where the SNP is or the variant is that we want to sequence. Okay, so that's the first thing I have my customer do. Give me the reference genome, give me the targets. And the first thing that I do when I go to design a targeted sequencing panel is I align all of the flanking sequences that they gave me. If they said this target is at position 170,000 on chromosome 1B, I map that actual flanking sequence that they get me back to the reference genome. And I double check that it's actually there and that it's accurate. Okay. And then I basically go into, and within, about 16 hours, I have a lot of Python scripting written and things that I do that allow me to design that panel. And then I go to Thermo Fisher, and then they manufacture the amplicons, the oligos, the forward and reverse primers to make the amplicons for that panel. And then we run it through a targeted sequencing workflow at the genotyping lab. Okay, so I hope that wasn't too much, but hopefully that was enough information that's what amplicon sequencing is. Okay, there's also Twist Biosciences and other companies out there that you may hear about that do target enrichment or hide capture. Okay, so that's a different approach, but it's all kind of within targeted sequencing because you are, even with target enrichment, you're basically focusing on specific targets in the genome that you're hybridizing oligos to amplify those, to bring those to light, and then to sequence those. These workflows, although they can be a lot more expensive because you're basically the investment is not only in the sequencing chemistry, but it's also in the oligo manufacturing. Okay, so let's just say that I have a customer that wants to design a 5,000 marker amplicon, or excuse me, a 5,000 marker panel. The first thing I'll tell him is, do you realize that just the oligos for manufacturing that are going to cost $75,000 alone just for the 5,000 marker panel? And usually they say, oh, well, maybe we should go with 1,000 markers or 2,000 markers, right? So that's kind of where you're at with targeted sequencing. Now, with low pass and whole genome, the library prep is very, very straightforward. You're just digesting it with an enzyme and sequencing it. What you're doing with those two technologies, though, is you're relying a lot on bioinformatics support to align those sequences back to the reference genome. All right. So I hope that puts everything in context in terms of what sequencing is. And then obviously you have Sanger sequencing, which is very straightforward. Sanger sequencing, you're looking at 200, 300 base pair amplicon or longer. And then you're just using the Sanger sequencing protocol to get all of your oligo sequences from that entire amplicon. All right. So it's very simple and straightforward. It's also very accurate. It's just a little lower throughput. It's not quite as high throughput as some of these other sequencing technologies. And then I put microarrays on this slide because although it's not sequencing, it is genotyping. And when clients want to discover, like I just talked to a client today who works in rice, and he says, Grant, we take about 10 of our best parents in our breeding program, and we do whole genome sequencing on them every year. Well, it's going to cost about $60 a sample to whole genome sequence them. And then from there, they go to a 2,000 marker targeted amplicon sequencing. So he was asking me, what's the pricing for you to design a 2,000 marker panel and take us from whole genome? And then can you help us with extraction, DNA extraction from whole genome sequence for a whole genome sequencing project? And I'm like, yes, yes, yes. Okay, what's it going to cost? So that's kind of how my business works, right? So, and then the microarrays basically are the ability to manufacture. And I always talk about microarrays as they're like a library book. If you've got 50,000 targets, which is a lot of targets, it's way too many for amplicon sequencing. We go up to about 5,000 targets for amplicon sequencing. Uh, we can take all those 50,000 targets or SNPs or variants, and we can put them onto a microarray, which is actually manufactured in Singapore at Thermo Fisher. Illumina has microarray technologies as well that are bead-based, but it's very similar. But those are there and they're manufactured, they're designed, takes about 12 weeks for the design process, but it can be a very good resource. And our customers, typically they will navigate between these four sequencing technologies. All right. So there, maybe I'll stop because I just gave you a lot of information and ask if there's any questions. Renuka, any comments?
No, Grant, it's beautifully laid out. The
Okay.
students, if you have any questions, anybody? So I have one question to ask you.
Mhm.
Grant, so the when we were remember at Devon Pole's lab, they had this sequinome, right?
Yeah.
Sequinome. So how does it differ from sequinome target? It's also we are targeting the SNPs. So what is the Pacific Coast genome? What's the difference basically? It's cost, yes, yes, of course.
Well,
Cost wise.
so Pacific Coast Genomics is we are just a service. We don't have our own sequencers per se. I do
Okay,
have my own
okay.
qPCR instruments, but I don't. So we're just a service. We're providing design. We do a lot of dry lab work, right, where we do the
Yeah,
bioinformatics
yeah,
work for the
yeah.
design and all of that sort of thing. Does that make
So now,
sense?
okay, yeah, that makes sense. So the you are mainly prefer towards which one? Like it depends on the, of course, customer need. But let's say you need uh, like millions of snips at least, like
Yeah.
let's say thousands of snips. So which one you guys are asking the customer
Right.
to go?
That's really good. Yeah. So when a customer comes to me, they say we need 1 million SNPs. I'll say, okay, we need to direct you to whole genome sequencing. This is the lab that can do whole genome sequencing for you. And then I go to that lab and I say, hey, can you give me a quote for running 96 samples for whole genome sequencing per sample? And they give me that quote, and then I send them to the lab, and the lab does the work. That's kind of how my business works.
Okay. Very nice. Yes.
Um, I will tell you though, you did bring up Devin. So Devin is actually one of my customers, and while I was at Thermo Fisher, Devin and I designed a 5K, a 5,000 marker wheat panel for the wheat group that they're still using today. So I still support them with Pacific
Oh,
Coast Genomics. I support them. We designed a new spike in a few weeks ago. I think I added about 50 markers to it. And so I still do that design work, and then I go to Thermo and order the oligos, and then I have them sent to the lab so Devin does not do any more sequencing anymore in his own lab
mm-hmm.
because the cost is so high.
Yeah,
So a
yeah.
lot of this is outsourced.
So those who don't know about, I mean, it's about a sequencing facility that in the Pacific Northwest, right? So
Yeah,
the
a lot
lab
of
that
them
we
are
are talking about for the students, I'm telling
yeah.
those who are not familiar, of course, this is a facility that in the Pacific Northwest they had the facility. Then what they are doing now collaborated with Dr. Grant as well.
Yeah.
So I have one more question. So you always have this the we need the reference sequence, right? What if the
Always.
reference sequence has mistakes? Like we can't do much, right? Do you? I mean, we really don't know if the reference genome is good or not, isn't it?
Well, a lot of times we learn over time whether or not the reference genome is good or not. That's a really good question, Renuka, because I oftentimes have to reconcile that with my customers. All right. So let's just give a perfect example. I had an onion customer he was working with an academic. He was in his own private company, but he came and said, I want to design 1,000 marker panel. I said, okay, send me your flanking sequences, which basically I asked for 200 base pairs upstream and downstream of the SNP where it's supposed to be. And then I asked them for the reference genome. And so he gave me both of those things. And then what I did was I mapped all of the flanking sequences to the reference genome to see if they mapped. There were about 400 that didn't map.
Wow.
And so I had another call with him, as I typically do. And that is what I call in my process the pre-designed QC process, where we're checking the quality of targets and how well they map to the reference genome. And I said, sir, we've got about 400 markers that aren't working. What do you want to do here? We can drop them from the design, or if you have confidence that the reference genome is not accurate, but these will amplify in your breeding material. We can go ahead and design the oligos, but it's it's going to cost us money to design 400 oligos if we decide to move forward. And he said, yes, we want to move forward. We want to go ahead and design those oligos. Well, we designed them and they didn't amplify. Now, the 600 that mapped well to the reference genome amplified great, but the 400 didn't. And so we had another call and he said, oh, yeah, I just checked some of those and they have errors in they have errors in them. So that was a case of where, we did the pre-designed QC and up front, I let the customer know these 400 markers are not good. I think we should drop them. But I gave him the ability to make the decision. Um, you know, but that's a perfect example of what you're talking about, Renuka, because the quality of the reference genome really matters. Now, in this case, the reference genome was actually pretty robust because it told us up front that we had about 400 targets that weren't mapping well, and we should have just, in hindsight, we should have just left those out, right? But that's real world. Yeah.
Yeah, yeah. Thank you. Thank you very
Yeah.
much.
Uh-huh.
Students, you can ask anything.
Excuse me, I also want to ask question. By the way, I am hi, Grant. So I'm a colleague of Renuka as well. So I just want to thank you so much for the insightful talk, and your journey is so inspiring.
Yeah.
So I just want to ask, like, what skills do you think students should develop to be competitive in the agri-genomics industry? Let's say crop improvement.
Yeah, no, that's great, Dina. So I'm really glad you asked that question. Thanks for speaking up too. And please, anybody else, if you have questions, I really want this to be, I don't want this to be a session where I give you a bunch of information that's not useful. So I've had students that call me from Stanford or other universities that are actually family friends or friends of people that know that I'm in this space and they're studying, right? So they're in their undergraduate years. There's two that called me that come to mind, and they're asking me the exact same question. So that's a really good question. And that's a good question to ask, especially if you're in your junior or senior year, right? And you're starting to think, okay, what's the next step for me? And a lot of times I will tell them you have two routes to take. Most people, if they work in a lab, they'll learn the wet lab. All right. But there's not a lot of money in the wet lab. It needs to be done and it's routine. And I do it. I do it because I need to amplify DNA and I need to extract it and but it's a utility. All right. So the real, I do have some slides coming up where there's other things that come into play, like interest level, right? Skill level, what you really want to do, because you need to be doing what you love, right? Not everybody loves bioinformatics, but where I'm going with this is the money is really where bioinformatics is because it's such a specialized need. And I do it because the need is there and there's not a lot of people doing it. To be honest, it takes a lot of attention and skill. In order to do bioinformatics, and anybody that I talk to that does it, you almost have to go to a room and get in a zone with full concentration because you're writing Python scripts and you're doing a lot of things that just require a lot of in-depth computational biology, right? So it's a certain skill set that you need. But I would answer your question saying if you can do that, if you have an interest in that, do it. A lot of jobs that I see, a lot of job announcements and that kind of thing, bioinformatics are paying anywhere between 180,000 and 220,000 a year. If you move up and you get into a real skilled level of being able to do that. Does that answer your question?
Yeah, thank you so much.
Sure. Yeah, and again, guys, ladies and gentlemen, ask questions because again, I don't want this to be a one-way delivery of information. I think this is a really good time, though, to talk a little bit about the historical context of sequencing, how this all fits together. I alluded to it a little bit in the inception of Illumina around 2006 that you saw change the pricing of sequencing over time. And so this was what kind of some of those first sequencers looked like. This is a picture of a MiSeq. And a lot of people beginning around 2006, 2007, they started doing whole genome sequencing, which I just spoke about in a previous slide. What they were doing with whole genome sequencing, and if you go to NCBI is it's the National Center for Biotechnology Information. And I mentioned to Deanum that dry lab work is in demand, bioinformatic work is in demand. If you're doing that kind of work, you really need to get to know NCBI well. So NCBI has stored in it crop genomes of multiple versions that have been released and published over the years. So for example, if I go to a paper and I have a client that wants onion SNPs, I'll go to a paper that was published, let's just say it was published in 2018, and they actually, a lot of times they'll give you the SNP information of, whole genome sequencing if they discovered 16,000 SNPs or 20,000 or whatever it was. And then I'll try to figure out the reference genome that they anchored those positions to and where they're at. And then I'll go to NCBI, I'll download the reference genome, and then I can do all my mapping and design work that way, right? So when we go back to 2007, these reference genomes were not built. So if you wanted to do what I do with my business today, you couldn't because it didn't exist, right? So that whole genome sequencing information was so important for us to learn that. And a lot of it was shotgun sequence. You'll see shotgun sequence was basically, small 150, 200 base pair fragments that were sequenced and aligned. And they'll tell you how they aligned and built the reference genomes. But this work was really important. And it was happening between 2007 and around 2014. Around 2010, I like to put this in here. This was kind of my post Thermo Fisher months that I put this presentation together. But Life Technologies purchased the Ion Torrent, which was their first access to sequencing. Now, the IP of this technology was different than Illumina because it used a hydrogen-based base calling system that was different than what Illumina uses. Illumina chemistry is a bit different. So it does paired end sequencing, and it has different chemistry and different reactions that actually do the functional sequencing. So the Ion Torrent that was purchased by Life Technologies, they still have that technology today. And it's the foundation for all the NGS sequencing work that we were doing. Okay. As things have progressed, the Ion Torrent and one of the knocks against it, or one of the disadvantages to the instrument, was it could only do 130 million reads, which back in 2014 was excellent. But the technology's come so far that 130 million reads is nothing compared to the billions of reads that current Illumina sequencers or Singular Genomics or Element Biosciences sequencers can do. So the sequencing technology has surpassed what the Torrent could do. But when we were doing targeted sequencing, it really didn't matter because we were utilizing targets in the genome, and 130 million reads was plenty for a 5,000 marker panel. Okay. And then around 2010, Thermo Fisher actually purchased, in 2016, I believe it was 2016 or 2014 that the acquisition happened where Thermo Fisher purchased Affymetrix microarray technology. And so then Thermo Fisher had microarray, a basic microarray business at that point. And that's part of what I supported when I was there. But basically, I call this a library book because each one of those pegs represents a sample. So this is almost like a 96. If you see this little picture down here, it's almost like a 96 well plate. It's usually in a 96 well format. And on each one of those pegs where you see that red silicon chip, there's actually, this was our larger format. There's actually upwards of 400,000 oligo markers that we could put on one single peg. So the 8,000K wheat array basically had two 96 well plates, 400,000 on one plate and 400,000 on another. So every sample you could interrogate 400,000 SNPs. Well, that was really important in the evolution of things because in the early days, between 2010 and 2014, that was a cutting edge technology because customers could go from whole genome sequencing at 1 million variants and they could put 10,000 or 80,000 or 50,000 of them onto a microarray. And that was very advantageous. And microarrays are still used today. Okay. So then in around 2016, my predecessor who had hired me at Thermo Fisher kind of birthed AgriSeq out of what was called the Clinical Sciences Division of Thermo Fisher and brought AmpliSeq over to the agricultural application. We brought the pricing down. We adjusted the chemistry a little bit for plants. And basically that technology is what I was working with when I was at Thermo Fisher as well. So we were looking at 50 to 5,000 SNPs. All right. And then currently, right, as I mentioned, customers are navigating between these technologies. So they're going from whole genome sequencing to microarrays to targeted sequencing, and then qPCR, Cas, TaqMan assays, which is what I run in my lab as well. That's part of what I do at my business. So putting this in another light or looking at it from another angle, this is a large Illumina sequencer. They have a NovaSeq now that, again, does billions of reads. And you can interrogate millions and millions of variants with this, right? It's a workhorse. It's a powerhouse. We had the Affymetrix microarrays. Illumina had a version of this in their Infinium as well. They called it their Infinium. But you could look at tens of thousands to 800,000 markers at a time. All right. And they're still using this technology today. I have a customer that I'm extracting DNA for and sending samples in for this sort of an analysis. And then targeted sequencing on AgriSeq, which is what I mentioned, looking at 50 to 5,000 markers. And then single marker analysis, which I have this instrument. I do have a wet lab and I have this instrument in my lab. I do a lot of bovine type marker work and wheat and different crops for single markers. All right. And that's a very lucrative business. It's a very low cost way of once you have discovered the SNP and you know what SNPs are important and what SNPs are linked to your trait of interest. So Renuka was asking me if stripe rust and resistance in wheat was still important. Yes, it is. And we have two markers that we use now routinely, right? So once you get down to the markers and the actual nitty-gritty of, okay, what markers are important for what type of application, then you can make real progress, right? And then you go into what's called routine screening at a very low cost. So when we look at the cost of all these, out of whole genome sequencing, you're looking at getting millions of variants that you can classify. Usually the turnaround time is anywhere between 3 and 6 months, depending on the lab and the business the company that you're going to. The cost is anywhere from $60 to $100 a sample, so it's pretty expensive per sample. And then you move from there to the microarrays, and for an 800,000 marker or a 400,000 marker microarray, you're going to be a little bit higher, a little bit over $100, $250 per sample. Once you get down to a 384 format and you're looking at 35,000 or 50,000 markers, the pricing can go down to $40 or even below that. With targeted sequencing, we were anywhere between $10 and $30 a sample. And at this point, you're starting to get into routine screening category to where the market and customers are saying, okay, now you're getting down to where you're giving me 2,000 markers at $10, $20 a sample. You start running more routine projects, your volume goes up, and you're running more samples in that type of a system. And then almost everybody is running a lot of samples every year at 25 cents to 40 cents a marker per sample on qPCR. So that's the gamut of where my business operates. I use every one of these technologies to some extent or another. Even though I only have qPCR in-house, all of my sequencing work and my microarray work is outsourced. Okay, so I'm going to go into a little bit of detail around the targeted sequencing panel design. We're getting a little long here. We're almost an hour. So Renuka, do you want me to stop here or do you want me to keep going? What's your take?
We'll keep going, Grant. Yeah.
Okay, good.
Thank you.
Okay. I don't want to go too long. I don't want to be that guy. So basically, I'm going to take you now through a targeted sequencing design. What we're looking at, I've touched on this a little bit, but again, we're interested in a SNP. We have the customer who sends us a flanking sequence. He gives me this SNP in brackets, and then he gives me 200 base pair to the right and 200 base pair to the left. I map that to the reference genome, and I make sure that everything checks out. Okay, when we go to actually design this, we're designing the oligos in red. So we've aligned it to the reference genome. We've gone through the design process. I know that SNP is located at a specific location in the reference genome. I upload that to my portal at Thermo Fisher, and I start the design process. At that point, the design at Thermo Fisher gives me it doesn't give me the oligos, but it gives me the design. They don't release the oligos because those are proprietary. But it basically it tells the manufacturer in Pleasanton what oligos to manufacture, and usually within 2 to 3 weeks I have it drop shipped to the lab that's doing the sequencing work. And so the design process for me is pretty quick. I do it in just a few weeks. Once customers submit samples and start running samples, what they get out of this technology is not only the SNP call, but because the 200 base pair fragment is being sequenced, they get a lot of novel variant information. Okay, so in part of what happens in the sequencing process, everything's sequenced, and then the server, the data analysis software, aligns every sequence fragment that's barcoded, so it knows where it is and what it came from and what sample it's associated with. It aligns it back to the reference genome, and then it gives us these novel variants. And these are very important to our customers. So I always like to highlight, not only does it give you the SNP, but a lot of times I'll go into the novel variants and I'll characterize those novel variants for the customers. And sometimes we'll make a 2,000 panel. Actually, with these novel variants, we can make it call, we can have 2,000 amplicons, but we can make it call as many as 10,000 variants because of all these novel variants that we've discovered, right? So at that point, we have to decide, okay, are these variants important or are they not? How much is too much information? How much information do you actually need? So I talked a little bit about what I did at Thermo Fisher Scientific, and this was one of the efforts that I led while I was there. So my whole theory while I was there, I was working for the microarray, the Axiom microarray business and supporting their globally as a product manager. But I also started to support the AgriSeek business because I really felt like we weren't penetrating certain markets in the globe because there were customers out there that weren't like Devon C that had access to SNPs and had done his own sequencing and all that. There were customers out there that would use our technology, I felt, if they had panels designed for them already. Okay. So I basically went to our bioinformatics team and I said, I acted like a customer and I brought them variants from customers that I would bring in. I would go to talk to the soybean group at the University of Missouri, or I would talk to the wheat group at Washington State University, or I would talk to the strawberry group at University of Florida and University of California, bring them in and we would design these panels. So in about, I don't know, less than a year, we designed these 21 panels that you see in this table. It happened fairly quickly. And those are all of the different customers that we worked with and the different panels across the different crops. And this was right before I ended up moving on from Thermo Fisher. But this effort, started to generate a lot of interest and revenue, a lot, and it started to revolutionize what we were doing with the technology at the time, and taking something very complicated and trying to make pre-designed panels so that people didn't have to go through the design process. It really took off. Okay, so a lot of this I'm still doing now at Pacific Coast. I support customers that are still doing these projects and want to sequence from these catalog products. So I've talked a lot about the technology, the functionality of the technology, the bioinformatics behind the technology, the design, and all of that. How do we actually take all of that and how do our customers implement that into their breeding programs and what they're doing? And I've got a slide coming up here as Renuka asked me to talk a little bit about my journey. But one of the aspects of anybody's journey, right? Any of our journey is as we learn highly technical information that very few people in the world's population actually get to access. We're very privileged in what we get to access, right? And how do we take that and actually apply it? Okay, so we learn to learn while we're in college, but one of the things that I've had to learn since I left college is how to apply what I learned. So I'm always learning. That doesn't stop. But I think one of the things that really makes you successful in business, and one of the things that really makes things work with your customers is, and a successful business, is being able to apply these technologies. So as you're learning this, you're actually in the driver's seat because you are the leader in your field. You're the expert in your field. And if you go on to get a master's or a PhD, you're going to be the expert in that space. You're going to be the leading researcher that's going to publish new information that's never been published before, right? You're going to be leading at that point. And so how do you actually take that though and make it into a solution that works for customers at the end of the day? And so that's the business of biology, right? And I have another slide coming up that talks about the business of biology. But this is kind of what we do at Pacific Coast Genomics. So with all of my experience up until this point, when I was at Thermo Fisher, I got to go right into what I really had desired to do for several years before I got there. And it was kind of like I was living my dream. I was able to go deep into these technologies, and I was able to learn, and I was able to learn the business aspects of what our customers were needing, what they were wanting. And that's really where Pacific Coast Genomics came out of. It was birthed in, DNA extraction services was birthed in. Our customers weren't getting good quality DNA that they were sending to us. The pilot program basically helps me as I bring customer samples in. It helps me tell them what kind of an extraction do you need, at what cost point do you need, and what's going to give you the best quality data when you go to sequence or qPCR or whatever you're trying to do with that. The amplicon sequencing panel design all came out of me seeing the need for customers that either needed pre-designed products, which I continue to do at Pacific Coast Genomics, or to help them in their current panel design. And then the single marker business, basically, I recognize the need that every customer has in their breeding program to characterize and classify single markers and sometimes bring them from discovery through amplicon sequencing to a single marker. Okay, this is the marker that you need to run for your trait of interest every year at a low cost, right? So that's kind of where all the aspects of Pacific Coast Genomics came from, needs, right? And so as you go through college, as you go through your classes, as you learn, think about the needs. And I'll tell you that a lot of what I do in my wet lab, I actually was doing in my PhD. So the chemistries that I have, high nuclei and dinuclei, the high nuclei actually came from the basis of it came from my dissertation. I had written in my methods and materials what I had used to extract DNA, what the chemistry was, and I built on it. Everything that I do to troubleshoot in the wet lab, whether or not a marker is working or whether or not a DNA extraction is good quality or not, it's all done by PCR. I learned to do all that when I was in my PhD. I learned to troubleshoot when I was in my PhD. I learned when things are not amplifying, something's wrong. Something's wrong with the DNA, something's wrong with the PCR mixture, something's wrong somewhere. I've got to go back and correct it. I do that all day, every day. And so I think that's what any business is built around. So Renuka asked me to give a little bit of context about business and what I do. And I'd say that's the greatest thing that I do is try to meet a need, right? That every customer has, and they're willing to pay for that service. So what do we do at Pacific Coast Genomics? I partner with a lot of people from Illumina. I still partner with Thermo Fisher. I have calls with Illumina about right now. I'm working with on the right. I have service providers. I'm working with service providers to figure out what's the next targeted sequencing platform. How do we move from Thermo Fisher's platform to an Illumina platform, or how do we move to a singular genomics platform? We're looking at that right now. And so I interact across all of these, from the customers doing the breeding. I still interact with Bayer. I interact with Syngenta to some extent, but really interact with USDA at Devon C at Washington State still. So my customers kind of span a broad range. I work with the service providers who actually do the sequencing. I work with them to try to get good quality DNA to them. Sometimes the customer can extract good quality DNA, sometimes they can't. Sometimes the service provider has a sequencing issue, and I try to step in the middle and try to come up with feasible solutions. So I talked about this a little bit earlier, but there's two different things, and I know that somebody had asked this question. Wet lab work and dry lab work, there's two different directions that you can go, and the dry lab is always a great direction to go. And then I'm just going to give you some fun little quotes. Some of these are anonymous. They might be for me, but I'm not going to let you know one way or the other. But as you're learning, remember that what you learn, you become. Okay, so your blood, sweat, and tears in studying and reading the books and studying the notes and studying for the tests. Okay, my dad, part of the reason I'm here is because my dad really instilled it in me that I needed to go to the library from 7 to 10 every night and study. That was a functional thing that he did when he was in college. He ended up being a professor at a small community college later in his career before he went back to farm full time. But this, I think, is a very important thing that you don't realize when you're in college and studying, that all of those things you're going to use later on in your career. Okay. Your greatest struggles can become your greatest strengths. I remember calling my dad. I was my first year of my master's, and I had a statistics class, And I studied and studied and studied, and I could not score well on the exams. And it was very frustrating. I almost gave up. My dad just encouraged me not to give up. And later on in my career, I ended up in Australia teaching statistics to field scientists who basically didn't understand why they needed replication and why they needed all of these things that you do in statistics when you do field plot design, right? So your greatest struggles can sometimes become your greatest strengths. But I was able to speak to them in a language and teach this, and I really enjoyed it. I came to enjoy statistics. It was amazing. Learn to apply what you learn, right? So you learn to learn when you're in college, and you'll always be learning, but learn to apply that. And that's really what I do in my business today. And don't allow anybody else to define you. You're always gonna run into those naysayers. You're gonna have those people leaving negative comments on whatever you post, right? And don't ever let any of that get into your head, right? So maintain your own confidence, integrity, strength within yourself to become who you want to go and really define your path. This is all an ongoing process, right? So everything that you're doing now in college, everything that you're learning, all the struggles that you're going through, it's all going to work out. It's all going to work for your benefit, and it's all going to be as you go. You're learning to, when you lift weights, you're lifting against gravity. You build muscle. It all works to your benefit, right? And then I want to leave you with some great quotes from Steve Jobs. Two people who I am inspired by are Steve Jobs, which you all know Steve Jobs, and Kelly Slater, who was a world champion surfer. Um, but Steve Jobs basically said innovation distinguishes between a leader and a follower. And keep in mind that you're in college right now, and that less than 7% of the global population has a college degree. Okay, so that gives you a leg up. That gives you innovation. That gives you that ability to do something with what you're learning, right? So the only way to do great work is love what you do. If you haven't found it yet, keep looking and don't settle. And I think this goes back to one of the students that was asking the question around, uh, the bioinformatics bit that I kind of went into there. But you kind of have to find what you love. If bioinformatics isn't what you love, but that's where the money is, go where you love. You want to be. Go to that subject matter that you love to do, that you enjoy. And just like Steve Jobs said, just like he said, if you haven't found it yet, keep looking and don't settle. And then he also said this, and I will tell you, I'm learning this. I've just learned it this week. I am as proud of what we don't do as I am of what we do. Innovation is saying no to 1,000 things. So stay focused and stay focused on what you love doing, and you'll be successful. So I'll leave it there, Renuka, and you can take it from here. I'm sorry that was a little bit long, but thank you, all of you, for joining today, and I hope that this was productive. Renuka has my information, and always reach out with any questions that you may have. My business is here to support people, and my advice is free. So feel free to reach out anytime. So.
Thank you very much, Grant. It's really, really inspiring, and it's once in a rare opportunity that our students get to listen to you. You had a big audience, like 70. Some had to go for a class at 9 o'clock, I think. That is why some of them left. But I get several messages that it's really inspiring. Kushani or anybody, I will talk to you, Grant, later. I know it's really midnight for you. Students, you may say something. Thank you so much. I highly appreciate it.
Good.
Dr. Poole, yeah, it was a really interesting session today, and thank you so much for taking your time to be with us despite your busy schedule, sir, and for sharing your valuable experiences and knowledge with us. And during the webinar, what came to my mind was the facts that you discussed regarding the sequencing market were somewhat new to us because although we study the concepts in theory, understanding how it operates at a commercial and industry scale was truly valuable, sir. And also, as students, it was really encouraging for us to hear from someone who has successfully connected science, technology, and entrepreneurship together. Thank you so much, sir. And it was really nice having you with us. Thank you.
Great. Thanks, Shirisha. Thank you.
Actually, sir, this was a really nice experience for us, and I have a question for you. For the student interested in genomics and biotechnology, what skills would you recommend focusing on if we want to move on to the sequencing or biotechnology industry?
That's a great question. So again, I'm going to go back to bioinformatics. Learn as much bioinformatics as you can. Get trained in Python. Um, those skill sets will set you apart. And you're always going to learn the wet lab work. Learn that as well. But really try to dig deep into the computational biology and learn as much as you can while you have the time to do that now.
Thank you, sir.
We are truly grateful for the time and effort you are dedicated to being with us, sir. Behalf of everyone present here, thank you once again, sir. It was an honor to have you with, sir. Thank you very much.
Great. Thank you.
Thank you so much, Dr. Poole, for that insightful and engaging presentation. On behalf of the Society of Plant and Molecular Biology and the entire department, I want to express our deepest gratitude to you. We know how demanding your schedule is, so we truly appreciate that you're taking your time to share your valuable knowledge and expertise with us today. So it has been a wonderful learning experience for all of us. And also, I would like to thank everyone in the audience in the morning and for active participation as well. A special thank you for Professor Renuka and Dr. Thilina for their support to coordinating the setting up for today's session. With that, we will officially conclude today's lecture. Thank you all once again and have a wonderful rest of your day.